Hello students, with the next topic, reversible and irreversible inhibition. We start talking about the regulation of enzymes, which was one of the most important characteristics of enzymes. First, there are two types of enzyme inhibition, non-specific inhibition that we talked about denaturation, about the pH optimum, temperature, heavy metals, reducing agents, and specific inhibition, which include irreversible and reversible inhibition. The reversible inhibition are competitive, non-competitive, and there is one more type, uncompetitive, but most often are the first two types. We start with irreversible inhibition. What is the most important feature of irreversible inhibitors? The irreversible inhibitor binds covalently and permanently to the enzyme. This is the difference, unlike the reversible inhibitors. Covalent bond formation there is between inhibitor and enzyme. And in the end, the effect is a completely loss of enzyme activity. The increasing of substrate concentration does not change the inhibition. You'll see different examples. Let's see the examples. Example number one are poisons like organophosphates. They irreversibly inhibit acetylcholine transferase by covalently modifying its active site. Result: neuromuscular paralysis, life-threatening toxicity. The second example is avidin. Avidin is one inhibitor which is found in the egg, in the raw eggs, and inhibits the biotin irreversibly. It's vitamin B7 or H, vitamin H, and cannot be absorbed after that. This leads to functional inhibition of all these enzymes that are biotin-dependent enzymes. Irreversible inhibition, the next example with aspirin. Aspirin, the other name is acetylsalicylic acid, is used for irreversible inhibition of the enzyme cyclooxygenase, which catalyzes the reaction from arachidonic acid to prostaglandins. COX, or cyclooxygenase, is inhibited irreversibly by a process called acetylation. The mechanism of action of aspirin is irreversible acetylation of hydroxyl group of serine residue in the active site of the enzyme. The inhibition can be for both of the cyclooxygenases, COX-1 and COX-2, by this covalent modification. The effect: inhibition of COX-1 is mainly associated with the antiplatelet effect or anti-aggregatory effect of aspirin. Inhibition of COX-2 is more important for anti-inflammatory and analgesic effect of aspirin. Since platelets do not have a nucleus and cannot synthesize new cyclooxygenase enzyme, the effect on them lasts for the entire life of the platelets, or around 10 days. The next example is penicillin. It's a so-called suicide substrate. Specific substrate analogs that enter the active site of the enzyme and bind covalently and irreversibly is this mechanism of action of penicillin. This is the structure of penicillin, this one, and this reactive peptide bond of beta-lactam ring is very important. Exactly after the degradation of these bonds, the penicillin is contacted with again two hydroxyl group of serine residue of the enzyme called glycopeptide transpeptidase. This enzyme is located in the bacterial wall. This is the action of penicillin. Again, will be the complex catalytically inactive, stable, and irreversible. Reversible, competitive, and non-competitive inhibition. Competitive. When we're talking about competitive inhibition, students, the first important is competitive inhibitor is structural analog to the substrate. This is the most important. Everything after that are consequences. They are structural analogs. That's why the only place of contact with the enzyme is only one. It's an active site. Why? Because they are the same. Second consequence. The complexes that are formed are only two. If there is contact between enzyme and substrate, enzyme-substrate complex will be formed in the end. If there is enzyme-substrate complex, product will be produced in the end, obligatory. But if there is contact with inhibitor, will be produced enzyme-inhibitor complex only. Is it possible to form enzyme-substrate inhibitor complex? No, because there is only one place of contact, the active site. Another question, is it possible this inhibition to be overcome, to be eliminated? Answer, yes. It could be, because if we have more substrate concentration, the substrate will be contacted with the active site. If there is more inhibitor concentration, the inhibitor will be contacted with the enzyme. That's why the name is competitive, which means competition. Competition between substrate and inhibitor. About enzyme kinetics. About enzyme kinetics, you can see this plot of Limeauer and Burke. Km increases with inhibitor, Km is increased, but Vmax doesn't change. Why Km increases? Because you know that the significance of Km means what is the affinity to the substrate. In this situation, increase affinity means decrease, increase Km means decrease affinity. High Km means that the affinity between enzyme and substrate is decreased. One more about this type of inhibition, competitive inhibition, is when we're talking about the pharmacological control, because we need to study such of these inhibitors because of the drugs. They are produced either like irreversible inhibitors, or like competitive inhibitors, or like non-competitive inhibitors. If we're talking about this type of inhibitors and using of drugs, we need to have very high doses of these drugs, because there is a competition with the natural substrate of the reaction. That's why the doses are high in this type. Okay, I said about that. Examples. The first example is malonate. Malonate is a structural analog of succinate and can inhibit the succinate dehydrogenase enzyme. This enzyme is called succinate dehydrogenase and cannot be produced the product, which is fumarate. This is the first example. The application of these competitive inhibitors, they're used as antimetabolites in the production of drugs against bacteria, viruses, or tumor cells. Example, okay, one by one you can see the examples, but before that you need to know about the toxicity that cannot completely spare human cells, because most pathways are common. They have specific selectivity, relies on differences in metabolic rate or enzyme specificity. And look the first example, students. Puromycin is a toxic drug, not used therapeutically. It's a structural analog, this is the puromycin, is a structural analog of tyrosine tRNA and inhibitor of protein biosynthesis. You can see very similar structure of this drug. The next example is acyclovir, this drug, which is structural analog of which base is, or which nucleoside is very similar to this drug. Which is this base? This base is guanine, and but no pentoses. The pentoses is not the same, and this drug is used as purine analog. This one is used as a pyrimidine analog because the base, only the base, is thymine. It's thymine. But in the pentose, instead of hydroxyl group here, there is one nitrogen atom. Azido 3'-deoxythymidine is the name. It is thymidine analog and inhibits the viral reverse transcriptase enzyme, interrupts the DNA synthesis because they are involved in the DNA structure of the viruses. Sulfonamides, this is the example of sulfonamides, are structural analogs of para-aminobenzoic acids. The mechanism of action is inhibition of synthesis of folic acid in bacteria, and bacteria cannot absorb folate from the environment. Because they must synthesize it. Look the scheme directly. The first part of scheme occurs only in the microorganism, and the second part in the humans, which means that in the microorganism the folate can be synthesized. If there is mechanism to stop synthesis of folate, or folic acid is the same, no folic acid for the microorganisms. This is the action of sulfonamides. They are used as competitive inhibitors to stop the reaction for the formation of folic acid in the bacteria because they are structural analogs of para-aminobenzoic acid. Methotrexate is the next example in this scheme, which is used as competitive inhibitor of the enzyme dihydrofolate reductase. Why? Because they are structural analogs of dihydrofolic acid or dihydrofolate. You can see the structure of methotrexate. This is the methotrexate structure, and this is the folic acid structure. Actually, for us, folic acid is a vitamin, B9, and we can accept this vitamin only with the food. But for the activation of folic acid, need to be activated to dihydrofolic and tetrahydrofolate thanks to two reductase reactions together with NADPH. The methotrexate application is to using in the childhood leukemia, also used in some autoimmune and malignant diseases. The other example is allopurinol. Allopurinol is a drug which is used for the treatment of gout because it is a structural analog of hypoxanthine. The last two reactions in the purine nucleotide degradation are these ones: hypoxanthine, xanthine, uric acid. These are the last two reactions. Both of the reactions are catalyzed by the same enzyme called xanthine oxidase. Allopurinol, because it is a structural analog of hypoxanthine, is an inhibitor of xanthine oxidase and no production of uric acid in the end. Actually, students, the gout can be treated by different mechanisms. One of the mechanisms of treatment is to reduce the formation of uric acid. You know from the previous lecture we said something about the defective enzymes which are the reasons for gout, but this is the treatment of gout with allopurinol. Here there is the general action of allopurinol. This is the enzyme xanthine oxidase that is inhibited by allopurinol, as I said before, but there is one more action of allopurinol to inhibit the main regulatory enzyme in the purine de novo synthesis, which is called PRPP amidotransferase. Students, look this dashed line. Up, all this is called de novo synthesis of purine nucleotides from ribose 5-phosphate to end products AMP and GMP. After that, start the process of degradation of purine nucleotides. In the end, uric acid is produced. Allopurinol is used by two mechanisms. Mechanism number one, competitive inhibition of xanthine oxidase. Mechanism number two, inhibition of the main regulatory enzyme in the de novo synthesis of purine nucleotides. Another example is ethanol, using of ethanol in some poisoning. For example, ethylene glycol is a component of antifreeze. Actually, it's not toxic itself, the ethylene glycol. The toxic is the product, which is oxalic acid, which leads to damage the kidneys and formation of insoluble crystals. If there is poisoning of ethylene glycol, ethanol can be used as an inhibitor of alcohol dehydrogenase, because this enzyme can use one more substrate, which is ethanol. Why not? Because they have very similar structure. That's why if there is poisoning of ethylene glycol, ethanol is used to stop the inhibition. One more, the same mechanism of action is with methanol poisoning. Methanol poisoning can be overcome with ethanol with the same mechanism. Non-competitive inhibition. We start with the most important feature of these inhibitors. They are not structural analogs of the substrate. No, they are different, with different structure. That's why there are many consequences. They cannot be attached to the active site. They are contacted with another site, maybe allosteric site, which means because there is another place of contact, can be formed all these three complexes students. If the contact is between enzyme and substrate, enzyme-substrate complex will be formed, which means obligatory after that product and unchanged enzyme in the end. If there is contact between enzyme and inhibitor, enzyme-inhibitor complex will be formed. But in this situation, because there is a free place for the contact with the substrate, some substrate can be contacted too. That's why it can be formed one new complex called enzyme-substrate inhibitor complex, unlike in the previous one inhibition. The same question, is it possible to be overcome this inhibition, to be eliminated? Answer, no. Why not? Because there is another place of contact with this inhibitor. If we're talking about the anti-metabolites or drugs, here the doses will be low. We don't need high doses of the drugs, because there is another place of contact, that's why. About enzyme kinetics, the changes will be in the Vmax. Line-Weber-Burke plot shows that Vmax decreases, but Km doesn't change. Why Km doesn't change? Because students, there is contact between enzyme and substrate. There is special place, active site is free to attach the substrate. That's why the affinities doesn't change. But the result, the total velocity of the reaction will be decreased because there is inhibitor which is contacted with the enzyme. Okay, that we said about that. This table shows the comparison between both of the inhibitors, competitive structural analog, non-competitive, not structural analog. Can be eliminated, cannot be eliminated. Activation of the enzymes. The definition: molecules that enhance enzyme activity and accelerate the reaction. These compounds are called activators. Type of activators: metal ions can be like magnesium, zinc, They facilitate or stabilize enzyme conformation. They can be different metabolites as, be careful, product inhibition and substrate activation. I will repeat again. Enzyme precursors, you'll see the mechanism of formation of active forms from the precursors, and allosteric activators. Okay, let's see one by one. First mechanism is formation of active forms of enzymes, but from proenzymes or pre-enzymes. These forms that are inactive are called zymogens. They are called zymogens. Which enzymes need to be activated and which enzymes are produced in inactive forms? These enzyme students are from the digestion. All digestive enzymes are produced in inactive forms. Why? To protect the tissue that synthesizes them. That's why. For example, pancreas, to protect pancreas, because the place of synthesis is one, pancreas, the place of action is another, in the small intestines, in the lumen of small intestines. The second examples are all enzymes that are involved in the blood clotting. Blood clotting, the other name is hemostasis. This system is used for one reason, to stop bleeding. All enzymes that are involved in this coagulation cascade are formed as precursors, as proforms. They are inactive normally. They will become active after their activation to stop bleeding commonly. Examples we'll see in the next slide. And here is written actually, prothrombin, thrombin, for example. In the digestion, pepsinogen, pepsin, trypsinogen, trypsin, or procarboxypeptidase, carboxypeptidase, proaminopeptidase, aminopeptidase, in the digestion. In fibrinolysis, this means that after the process to stop coagulation, this plot, this coagulum need to be destroyed, the fibrin need to be degraded by a process called fibrinolysis. That's why here is written fibrinolysis. The example is plasminogen, plasmin. The process of activation of these forms is called limited proteolysis. Limited proteolysis. Here there is activation of the enzyme precursor trypsinogen into the active enzyme trypsin. After this process of autocatalytically change of the polypeptide chain by cleavage in some part of the polypeptide chain, and after that the active site can be formed. The next example is formation of thrombin from prothrombin. This is the prothrombin from the cascade of coagulation. Actually, this is the factor 2 from the cascade of coagulation, and thrombin is factor 2A, which means active form of thrombin. Here, there is only one polypeptide chain, and after these two cleavages of process called limited proteolysis, two polypeptide chains are formed with one interchain disulfide bridge, and thrombin is active now. Allosteric control. Allosteric control refers to allosteric enzymes. Allosteric enzymes are these enzymes that have allosteric site. All enzymes have active site, but not all enzymes have allosteric sites. Only these enzymes can use such a dysregulation with allosteric regulation. Allosteric effectors bind there. They are called allosteric activators or allosteric inhibitors. They have no structural similarity to the substrate. That's why this allosteric inhibition is similar to noncompetitive inhibition of the enzymes. After the binding with activator or inhibitor, after that there is conformational changes in the active site, and in the end the result will be increase or decrease activity. Also, all this, the regulatory and catalytic subunits can be different. One more, all allosteric enzymes are with quaternary structure. Also, they are regulatory enzymes, all allosteric enzymes. The classical example of allosteric regulation are the synthesis de novo of purine and pyrimidine nucleotides. The next, the Michaelis-Menten kinetics is not valid here. They do not follow these kinetics. You understand about the Michaelis-Menten kinetics, which shows the dependence between enzyme reaction rate and substrate concentration. In non-allosteric enzyme, this dependence is hyperbolic. But here, the dependence is not hyperbolic in allosteric enzymes. The dependence is sigmoidal curve. There is a dependence instead of rectangular hyperbola. And the reason is because of the cooperative effect between subunits. Because quaternary structure means more than one subunit, more than one polypeptide chains connected between each other with non-covalent bonds. A retroinhibition as a type of allosteric inhibition. Students, in our organism there are two types of reactions. The first types of reactions are circular reactions, like Krebs cycle, urea cycle, but all the rest are reactions in order, one by one, and for example, 10 reactions or 20 reactions, never mind. But one of the reactions is the main important, is the main enzyme. Which is this the main regulatory enzyme of the reactions? This is the slowest reaction. That's why this reaction is called rate limiting step. This enzyme is the most important. Everything about the activation or inhibition of whole pathway depends on this enzyme. That's why here is in general scheme, which shows, for example, enzyme number 2, which can be inhibited by the end product. Remember, students, the most important, most often inhibitor of the reaction is the product. That's why the name is product inhibition. The most important allosteric activator of the reaction is the substrate of the reaction. That's why here is written A activates the main enzyme, because this is the substrate of all this pathway. E is the final product, which inhibits the main enzyme. One more, each product can inhibit its own synthesis too. This is called product inhibition. Classical example with purine synthesis. We said many times about purine nucleotide synthesis. Look again. In this scheme, you can see the substrate of the reaction, ribose 5-phosphate, but PRPP is the active form of the substrate. That's why actually PRPP is the real substrate, because it is the active form of the substrate. Which are the end products? The end products are GMP, GDP, GTP, AMP, ADP, ATP. All these end products inhibit the main enzyme. Why? Because they are products. That's why they are the main inhibitors of the main enzyme of the process, which is the activator of this process. You can see this plus, which comes from PRPP. Why from PRPP? Because this is the real substrate of all this metabolic pathway. Actually, here there are two more pluses. This one, which activates the opposite process of synthesis of ATP, and ATP, which activates the opposite process of synthesis of GTP. This is called reciprocal activation, which is used especially in the purine and pyrimidine nucleotides. The reason is to have enough quantity of all nucleotides to be incorporated with the same possibility into DNA or RNA. That's why exists such this reciprocal activation. The next example is pyrimidine de novo synthesis. The same, absolutely the same is, which are the end products? The end product, you can see minuses. UTP, uridine triphosphate, inhibits. Cytidine triphosphate inhibits. Thymidine diphosphate inhibits. And which are the activators? Again PRPP, because this is the real substrate for both of the synthesis of pyrimidines and purines. And here is one interesting more activator, which is ATP, which is not pyrimidine, it's a purine nucleotide. But this purine nucleotide activates the pyrimidine synthesis. Why? The same reason like in this reciprocal activation of opposite nucleotides to have enough possibility to be incorporated into DNA or RNA synthesis. This example shows the activation from the substrate of the reaction. For example, it's a metabolic pathway from glycolysis pathway. Fructose, this fructose 1,6-biphosphate activates one of the final enzymes in the reaction called pyruvate kinase. And that's all from today's students.